Okay, today we're going to talk about human genetic mutations. There are two main types of mutations. First is the chromosomal mutation, which may affect parts of the chromosome or may affect several genes of the chromosome, and then gene mutations, which only affect a particular type of gene. So to remember that chromosomes are part of DNA and it's coiled up, and then it's usually what's used to divide in mitosis or meiosis. There are 23 pairs in humans, and we go from the largest to smallest when we do a karyotype, which is like the little box that you see over in the corner. And it's basically just a picture of all the chromosomes that a human would have so that you can compare and see if they're a normal pairing or if something is a little off. So chromosomal mutations, these are any change in the structure or number of chromosomes. It's a large scale effect, so it affects many genes. There are 5 different types. There's deletion, duplication, inversion, translocation, and non-disjunction. So for deletion, you have part of the chromosome is taken away. So in this case, C and D genes are taken away or lost. So there are several different causes that you can see. And so the first syndrome that you have here is a severe mental retardation or handicap. The Creutzfeldt syndrome has like a child making a meowing sound, and it usually results in a mental handicap as well. Inversion is where you may have genes that are flipped. So here you have C, D, and E, so it is flipped to E, D, and C. And so you have what's called the 4-ring syndrome. You have a cleft palate, club feet, and testes don't always descend, so sometimes they're sterile. You have translocation is the next one, and this is where you swap. So you have one chromosome swapping material with another chromosome, and sometimes that may leave sections of chromosomes missing. Non-disjunction, this is where chromosomes fail to separate during meiosis. So instead of one part of the chromosome, you have maybe all 4 parts of the chromosome. This produces gametes that may be missing chromosomes or have extra chromosomes. So you can see that here in meiosis 2, they basically have supposed to be one chromosome in each egg, and then they separate into one chromatid in each egg, and then this one did not separate, so this egg doesn't have anything. So when you have a fertilization with this, you end up with a trisomy effect where you have 3 different parts in your egg instead of 2. So every cell in the baby's body will have 3 chromosomes or 3 copies of the chromosome instead of 2, and this condition is called trisomy. Trisomy 21 is when an individual has 3 copies of chromosome number 21, and that's usually called Down syndrome. So most chromosomal mutations are lethal. If the fetus survives, they tend to cause widespread abnormalities. So this is like Down syndrome. So you have 3 chromosomes in the 21st pair for Down syndrome, and this would be what a karyotype would look like for a Down syndrome child. In this pair, you have 3 instead of 2 like you normally have in all the other ones. So you can do genetic screening for this before the baby is born. It's through a process called amniocentesis. Amniocentesis, they stick a needle into the uterus of the mother. They draw out fluid that surrounds the baby. Then they bring the fluid out, centrifuge it, and then they basically can take that and put it on a growth plate, and then they can do the cell culture and see if the cells are normal. Gene mutations, these are small scale. They're usually only affecting one gene at a time. Any change to the DNA sequence of a gene is going to affect by either adding a base, changing a base, or it could be missing a base. So there's 2 main types of gene mutations. There's what's called a point mutation, where only one particular base is changed. There's a frame shift mutation, which comes in 2 forms, either inserting a base, which causes everything to shift, or deleting a base, which causes everything to shift. So point mutations, one base is usually substituted for another. one of the things that can be caused by point mutation is sickle cell anemia. There are three possible consequences. There's three possible point mutation types. One is called a nonsense mutation, and this is a code for a stop, which can translate the protein in a different way. So here you have an insertion of a stop codon in the amino acids, which stops the protein synthesis, which may not allow the protein to be completely formed and have the proper use. There's a missense mutation. This codes for a different amino acid, and that can create a different protein, which has a different function. And then you have the silent mutation, which basically there's no change in the amino acid. So the amino acid might have a different base, but it pairs for the same amino acid, and therefore you don't seem to have a difference. The next type is frame shift mutation. And frame shift mutation is usually where you're adding or deleting a base, and that would be something like cystic fibrosis. So causes of mutations are spontaneous. Most are spontaneous. They don't have a set point at which they're going to mutate. This occurs during your DNA replication, and it's caused by mutagens. And mutagens can be lots of different things. They can be physical, like example would be like radiation from UV rays, X-rays, or extreme heat, or they can be chemical, which the molecules that misplace the base pair so disrupts the helical shape of the DNA. They can be environmental. It can be cigarette smoking can be a chemical that would change it. PCBs would be another one that could change it. So those would all be forms of mutagens. So the key idea is that a mutated gene will make a mutated protein. So mutant proteins are trouble. They do not go where they are supposed to go. They do not do what they're supposed to do. And so you have some situations there. So here are some examples. Example would be sickle cell anemia. So your normal blood cells, they are shaped like a disc. They have a hemoglobin, which carries oxygen to all parts of the body, and that's your protein. A sickle cell is shaped like a crescent, and so the hemoglobin is abnormally shaped. They don't move easily through your blood vessels, so they usually get clumped up together and start forming clots and can cause the blood not to flow properly. So if you look at the top, that's a normal red blood cell, and the bottom is a sickle cell, which is anemia. Cystic fibrosis, that is an autosomal recessive disorder. It's where you have thick mucus in your lungs and digestive tract. So you have like a constant lung infection and impaired digestion. Basically, 1 in every 3,000 Americans can have cystic fibrosis, and 1 in 23 white Americans can carry this particular allele. So basically, in cystic fibrosis, the gene is mutated, so you have 3 base pairs that are basically deleted. And since it's deleted, that mutant protein is missing an amino acid and it can't fold correctly. And remember, the way that a protein folds is what the function of that particular protein is. Some questions that you can go back and review.